Take a breath, not quickly, slowly. Let the air fill your lungs completely. Feel the chest expand. Feel the rib cage open. Hold it for a moment. Feel the oxygen crossing into your blood. Feel the faint warmth of the metabolic reaction that your body is running 24 hours a day. The reaction that turns the oxygen into carbon dioxide and produces the energy that powers every thought you have, every heartbeat, every quiet second of being alive. Now consider this. 2.5 billion years ago, that breath would have been an apocalypse. The gas you just inhaled, the gas that is so normal to you, so absolutely taken for granted, so unremarkable in its constant presence, was once the most dangerous substance on Earth. It was poison. It was catastrophe. It was the end of a world. Oxygen did not always exist in Earth's atmosphere. For the first 2 billion years of Earth's existence, more than half the planet's current age, the air contained almost none of it. The world breathed different gases: carbon dioxide, nitrogen, methane, hydrogen sulfide. And in that atmosphere, in those first 2 billion years, life had arisen, had diversified, had covered the planet in billions of invisible organisms, had filled the oceans with chemical energy and biological complexity, all without oxygen, all in conditions that would kill you in minutes. And then, approximately 2.4 billion years ago, a tiny, single-celled organism began producing oxygen as a metabolic waste product. It did not intend to reshape the planet. It had no intentions at all. It was a bacterium, a thing of pure chemistry and replication, without goals or awareness or any concept of consequence. But the consequence was the largest extinction event in Earth's history, greater in its totality than the asteroid that killed the dinosaurs, greater than the volcanic catastrophe that ended the Permian, greater than any mass extinction that has a name and a study and a chapter in the paleontology textbooks. The oxygen was the poison, and the oxygen was the gift. These two things are not contradictions. They are the same event, seen from two different sides of 2.4 billion years of time. The catastrophe that killed almost everything alive on Earth was simultaneously the only possible path to the world you are lying in tonight. Without the poison, no complexity. Without the apocalypse, no fish, no forests, no dinosaurs, no mammals, no humans, no one to lie in the dark and think about how extraordinary it is to breathe. Tonight, we are going to go back to the world before the poison. We are going to understand what was there, what extraordinarily diverse, extraordinarily ancient, extraordinarily alien living world existed before oxygen appeared. We are going to watch the poison arrive, slowly, inexorably, transforming everything. We are going to witness the slow apocalypse that no one could survive and almost no one could escape. And then we are going to follow the survivors through the 1 billion years of recovery, through the oxygen-fueled explosion of complexity that produced everything you know. And at the end, we are going to ask, what if it had never happened? What if the cyanobacteria had never evolved? What if the oxygen had never come? What would the Earth be tonight? Let your breathing slow. Feel the oxygen in the air around you, the oldest miracle, the youngest element in Earth's atmosphere, the substance that is doing your thinking for you, right now, in this quiet room, in this quiet night. Let us begin. What you are breathing right now, the air in this room is, by mass, approximately 23% oxygen, by number of molecules, about 21%, The rest is mostly nitrogen, with trace amounts of argon, carbon dioxide, water vapor, and dozens of other gases in quantities too small to matter much. You breathe this mixture approximately 20,000 times per day, extracting roughly a quarter of the oxygen from each breath and exhaling the rest, along with the carbon dioxide your cells have produced. The oxygen molecule, O2, two oxygen atoms bound together, is not a stable thing. Oxygen is, chemically speaking, a highly reactive substance. It attacks other molecules. It strips electrons from them. It oxidizes them. In the formal chemical sense, the same process that rusts iron, that turns a cut apple brown, that makes a fire burn. Your cells are running a controlled version of this same reaction. Controlled oxidation, carefully managed, the energy extracted in small increments and stored in molecules of ATP, the universal energy currency of biology. In your cells, right now, oxygen is attacking organic molecules with extraordinary precision and efficiency, releasing energy that powers everything from your heartbeat to your thoughts. This is aerobic respiration. It produces approximately 18 times more energy per glucose molecule than the alternative anaerobic respiration, which does not use oxygen. 18 times more. This difference in energy yield is not trivial. It is the difference between a cell that can barely maintain itself and a cell that can afford to power a complex organism, to run a nervous system, to build and maintain a body of trillions of cells, to grow a brain large enough to contemplate the nature of the very gas that is powering it. Oxygen is expensive. It is dangerous. Its reactivity means that it damages biological molecules if not carefully managed, and the management of oxygen damage is a significant metabolic investment that every aerobic organism makes. But the energy it provides makes that investment worthwhile many times over. Oxygen is the fuel that complex life runs on, but this was not always so. For more than half of Earth's history, no organism ran on oxygen. The world ran on other fuels, other chemical gradients, other energy sources. And those organisms, the anaerobes, the iron breathers, the sulfur eaters, the methanogens, were not simple or primitive in any meaningful sense. They were complex, diverse, ancient beyond anything we can easily imagine, perfectly adapted to a world that no longer exists. They were the world that oxygen killed, the world before the poison. 4 billion years ago. The Earth is 500 million years old. The surface has cooled enough to hold liquid water, though not always. Not everywhere volcanic activity is intense, and the bombardment of the inner solar system by leftover debris from the formation of the planets is still ongoing, each large impact briefly remelting the surface and reboiling the shallow oceans. The sky is not blue, it is orange-brown, thick with methane and other organic haze compounds that scatter light differently from the nitrogen and oxygen of the modern atmosphere. The methane in the Archean atmosphere is the product of biological activity. The methanogens, ancient archaea, that consume hydrogen and carbon dioxide and exhale methane as a waste product, are already at work, filling the atmosphere with their exhalations. The methane is a greenhouse gas, trapping heat from the sun at a time when the sun is approximately 70% as bright as it is today. Without the methane greenhouse, the early Earth would be frozen, the so-called faint young sun paradox. The methanogens are keeping the planet warm. They are maintaining the conditions for the very liquid water that their biology requires. The ocean is not blue either. It is greenish-brown, or sometimes reddish. colored by the dissolved iron that permeates the early ocean. The Archean Ocean is rich in ferrous iron, iron in its reduced, dissolved form, which can exist in solution only in the absence of oxygen. Where there is oxygen, iron precipitates as iron oxide rusts and falls to the seafloor. In the Archean Ocean, there is no oxygen, and so the iron stays dissolved, coloring the water, available as an energy source for the iron oxidizing bacteria that will use it as fuel. The smell of the Archean world, if you could be there to smell it, which you could not, because you would suffocate almost immediately, would be extraordinary. Sulfur, the thick, eggy smell of hydrogen sulfide wafting from volcanic vents. The sharper, more acrid smell of sulfur dioxide from the volcanoes themselves. The faint organic smell of the ocean surface, the chemical byproducts of 1 billion different microbial metabolisms, and beneath all of it, the smell of iron, the metallic, cold smell of iron-rich water, of rust before there was rust. This is the Archean Earth, an alien planet, a planet that we would not, if we visited it without prior knowledge of Earth's history, recognize as Earth. A planet that is, in its chemistry and its atmosphere and its ocean and its light, completely different from the world we know. And yet it is our world. It is this world, 4 billion years ago, and everything that exists today grew from it. Life is already here, has been here, perhaps, for as long as 200 million years already. The oldest evidence of life on Earth, isotopic signatures in ancient rocks from Greenland, carbon chemistries in Western Australian rocks suggests that life appeared astonishingly quickly after the conditions for it were established. By 4 billion years ago, life is here. It is small. It is invisible. It is chemistry wrapped in a membrane. But it is here, and it is already diverse, already running dozens of different metabolic strategies, already exploiting every available chemical gradient in the Archean Ocean and the Archean seafloor. This is the world that will be transformed by the poison, the Archean Ocean. Beautiful and deadly. To understand the Archean Ocean, you need to let go of every assumption you carry about what an ocean should be. The modern ocean is a place of photosynthesis and oxygen. The surface layer is sunlit, filled with phytoplankton running oxygenic photosynthesis, turning sunlight and carbon dioxide and water into organic matter, and crucially, releasing oxygen as a byproduct. The oxygen dissolves into the surface water and is mixed downward by currents and waves. The deep ocean, though less oxygenated, receives oxygen from the sinking of cold, dense surface water. The entire ocean is, to some extent, oxygenated. Anaerobic conditions, truly oxygen-free conditions, exist only in specific, restricted environments. The depths of certain stratified basins, the sediment a few centimeters below the seafloor surface. The Archean Ocean was entirely the opposite. There was no free oxygen anywhere in the ocean. The entire water column, from surface to seafloor, was anoxic, reducing, full of dissolved compounds that could react with oxygen if oxygen were present. Dissolved iron, dissolved manganese, dissolved hydrogen sulfide. These compounds existed precisely because oxygen was absent. They were the chemical inventory of a world that had never been oxidized. In this ocean, the chemical gradients that powered life were different from the chemical gradients of the modern ocean. Instead of the oxygen gradient that modern aerobic life exploits, the Archean Ocean offered iron gradients, iron-rich deep water, meeting iron-poor surface water, where any photochemical reactions might have created transient, oxidizing conditions. Iron oxidizing bacteria sat at this interface, using the energy of the iron oxidation reaction to power their metabolism. Sulfur gradients. The volcanic vents of the Archean seafloor produced hydrogen sulfide in vast quantities. Sulfur oxidizing bacteria sat at the interface between the hydrogen sulfide plumes and the slightly less reduced water above them, extracting energy from the oxidation of sulfur compounds. Sulfate-reducing bacteria did the reverse, taking sulfate and hydrogen, extracting energy, and producing hydrogen sulfide as a waste product, a complete sulfur cycle, entirely without oxygen. Methane gradients. The methanogens, perhaps the most ancient of all the biochemical lineages, the ones whose metabolism is considered closest to the metabolism of the very first cells, consumed hydrogen and carbon dioxide and produced methane. The methanotrophs consumed methane and produced carbon dioxide. Another complete cycle, again entirely without oxygen. Hydrogen gradients. Molecular hydrogen produced by volcanic reactions, by the weathering of iron-rich rocks, by certain microbial processes, was a fuel for the simplest of all metabolisms, the one that may have been the first metabolism of all. Hydrogen-consuming archaea extracted energy from the simple reaction of hydrogen with carbon dioxide. These are not primitive metabolisms in the sense of being simple or inefficient within their own chemical logic. They are ancient. Certainly, some of them may be as old as life itself, but they are complete, self-sustaining, self-replicating, chemically sophisticated in ways that took human biochemists most of the 20th century to fully work out. The organisms running these metabolisms are not lesser organisms. They are organisms perfectly adapted to the chemical world of the Archean. The Archean ocean was dense with them. Not visible to the naked eye, most of them are microbes, single cells a few micrometers in size, invisible without microscopy, but present in extraordinary numbers. A milliliter of modern seawater contains approximately 1 million microbial cells. The Archean ocean, with its rich chemical energy gradients and its lack of predation by any large eukaryotic organisms, may have supported even higher microbial densities. Across the entire Archean ocean, 4 times 10 to the 20th liters of water, the total number of microbial cells was astronomical. They were the world, the creatures that ruled the anaerobic earth. Who were these organisms? The masters of the pre-oxygen earth. They were 2 things, primarily: bacteria and archaea, the 2 domains of prokaryotic life that have been here since the beginning, that are still here now, that constitute the invisible majority of all life on Earth, even in the modern, oxygen-rich world. In the Archean, they were everything. The archaea, particularly the methanogens, deserve special attention because they may represent the oldest living metabolic strategy on the planet. Methanogens use an extremely simple chemistry. They take hydrogen, H2, and carbon dioxide, CO2, and combine them, extracting a small amount of energy from the reaction, and produce methane as a waste product. This reaction, called methanogenesis, may be as old as life itself. Some researchers argue that it was the first metabolism, the very first chemical trick that life discovered for extracting energy from the environment. The methanogens are extraordinary organisms. They live in conditions that would kill almost anything else, completely anaerobic, often very hot, often very saline, often under enormous pressure. They live in the deep subsurface of the earth. Drilling into ancient rock has found methanogens living in tiny fractures in rock kilometers underground, sustained by the hydrogen produced by the reaction of water with iron-bearing minerals. They live in the hot springs of Yellowstone, and in the sediment of marshes, and in the digestive systems of termites and cattle. They are ubiquitous, ancient, and almost completely indifferent to the conditions that would define hostile environments for every other kind of life. In the Archean Ocean, the methanogens were among the most important organisms. They produced the methane that warmed the planet. Without their methane exhalations, the Earth would have been frozen. The faint young sun would not have been able to maintain liquid water without the greenhouse contribution of the methane. The methanogens were, in a very real sense, the organisms that made the Archean Earth habitable, not for complex life for themselves and for the other anaerobic organisms that shared the ocean with them, but habitable, warm, liquid, alive. The sulfur-reducing bacteria were equally important. Their metabolism, the reduction of sulfate, so 4 squared mim, using organic carbon or hydrogen, producing hydrogen sulfide, H2S, as a waste product, is one of the most widespread and most ancient metabolic strategies in the bacterial world. In the Archean Ocean, with its volcanic sulfur supply and its reducing chemistry, the sulfate reducers thrived. Their hydrogen sulfide production was a significant factor in the ocean's chemistry, maintaining the reduced, anoxic conditions that characterized the Archean Ocean. The iron-oxidizing bacteria occupied a more energetically challenging niche. Iron oxidation releases much less energy than the metabolic reactions available to aerobic organisms. But in the Archean Ocean, where iron was abundant and oxygen absent, iron was the available fuel. These organisms sat at the chemical interface between iron-rich deep water and slightly more oxidizing surface water, processing iron one atom at a time, building their cellular machinery from the slim energy margin that iron oxidation provides. And then there were the anoxygenic photosynthesizers bacteria that had discovered how to use sunlight as an energy source, but through a different mechanism than the cyanobacteria that would come later. The anoxygenic photosynthesizers used hydrogen sulfide, or ferrous iron, or organic compounds as electron donors in their photosynthetic reactions, producing sulfur or iron oxides as waste products, not oxygen. These bacteria existed in the Archean Ocean, in the shallow sunlit zones, extracting the energy of sunlight without producing the dangerous byproduct that would later transform the world. The anoxygenic photosynthesizers are important because they represent an evolutionary step on the path to oxygenic photosynthesis. The machinery of photosynthesis, the light harvesting complexes, the electron transport chains, the carbon-fixing enzymes, was developed in these organisms. The cyanobacteria, when they appeared, did not invent photosynthesis from scratch. They inherited it and added one critical modification. They found a way to use water as the electron donor instead of hydrogen sulfide or iron. Water is enormously more abundant than hydrogen sulfide. Water is everywhere in the ocean, and the waste product of using water in photosynthesis is oxygen. That modification, that one critical, world-destroying innovation, was still in the future. In the Archean, the anoxygenic photosynthesizers thrived in the sunlit surface waters. The methanogens thrived in the reducing depths. The sulfate reducers worked the sediment, the iron oxidizers worked the chemical interfaces, and the entire system was stable, an ancient, self-sustaining, self-regulating world that had been running for 1 billion years and showed no signs of ending, iron breathers and sulfur eaters. Let us spend a moment with these organisms, not rushing past them as mere background, as the before picture in a before and after story. Let us understand them as what they were: extraordinary biological machines, refined by 1 billion years of evolution, perfectly adapted to a world we cannot visit. The iron-reducing bacteria organisms that use ferric iron as a terminal electron acceptor in their respiration, producing ferrous iron as a waste product, are in some ways the most ancient of all the iron metabolizers. They may have been among the first respiratory organisms, using the chemical gradient between different iron oxidation states as their energy source. In the Archean Ocean, with its abundant dissolved iron in various oxidation states, the iron reducers had essentially unlimited fuel. They processed iron 1 electron at a time, extracting the energy difference between ferric and ferrous iron, and building their cellular machinery from this modest but reliable energy yield. Modern iron-reducing bacteria exist today. Geobacter and Shewanella are the best studied examples. Geobacter can respire using solid iron minerals as terminal electron acceptors, essentially breathing rock. It transfers electrons through long, hair-like protein filaments called microbial nanowires that extend from the cell body to the surface of an iron mineral particle meters away. The bacterium sits in the sediment, reaches out its nanowires, and respires on iron minerals in its immediate environment. This is not metaphor. This is literal. A bacterium that breathes iron, that sends out wires to transfer electrons to rocks, that has been doing this, or something like it, for perhaps 3 billion years. The sulfate-reducing bacteria are equally ancient and equally remarkable. Their metabolism is the basis of one of the most important biogeochemical cycles on Earth, the sulfur cycle, and their activity is detectable in the geochemical record of some of the oldest sedimentary rocks on the planet. The sulfate-reducing bacteria take sulfate, an oxidized sulfur compound abundant in ancient ocean water from volcanic and hydrothermal sources, and use it as the electron acceptor in their respiration, reducing it to hydrogen sulfide. The hydrogen sulfide is toxic to most modern organisms. To the sulfate reducers, it is exhaust. The smell of hydrogen sulfide is the smell of rotten eggs, specific and unmistakable. It is the smell of marshes where organic matter is decomposing, of hot springs with high sulfur content, of the deep sea floor near hydrothermal vents. It is a warning smell. In the modern world, a signal that conditions are anaerobic and potentially dangerous. In the Archean world, it was the smell of life. The dominant metabolism of the seafloor was producing hydrogen sulfide everywhere, all the time, filling the bottom water with the rotten egg odor of active sulfate reduction. The methanogens complete the picture, their methane production invisible, odorless in small quantities, was pervasive. Every reducing environment in the Archean Ocean was a site of methanogenesis, the sediment of the ocean floor, the deep waters below the photic zone, the cracks and crevices of the sea floor where hydrothermal fluids mixed with the overlying water. All of it was generating methane, slowly, continuously, filling the ocean water with dissolved methane, releasing it to the atmosphere, where it accumulated to levels that would be toxic to the aerobic cells that would eventually evolve. These organisms, the iron breathers, the sulfur eaters, the methane makers, were not primitive in any sense that matters. They were exquisitely calibrated to their environment. Their enzymes were adapted to the specific chemical conditions of the Archean, the specific redox potentials, the specific temperatures, the specific concentrations of metals and compounds. They had been evolving for 1 billion years or more. They were the product of 1 billion years of selection, 1 billion years of optimization, 1 billion years of arms races with each other and with the chemical environment. They were the rulers of the pre-oxygen Earth, and they were about to be destroyed. Stromatolites, the architects of the apocalypse, walked to the shores of Shark Bay in Western Australia on a calm morning when the water is clear and the low sun is lighting the shallows from a low angle. Look into the water. On the flat, sandy seafloor, rising from the shallow water in rounded, dome-like columns, are structures that look like rocks, cauliflower-shaped, layered inside when you cut them open, each layer representing approximately 1 year of growth. They range in size from a few centimeters to 1 meter or more in height. They are slow-growing, patient, ancient-looking. They are the most important organisms in the history of life on Earth. They are stromatolites, and their ancestors built the world you live in, and in building it, destroyed the world that came before. Stromatolites are layered structures built by microbial mats, communities of microorganisms dominated by cyanobacteria that grow in thin films on surfaces in shallow, sunlit water. During the day, the cyanobacteria photosynthesize. They capture sunlight. They fix carbon dioxide. They grow. As they grow, they trap sediment particles from the water above them. The sediment accumulates in a thin layer on top of the mat. The cyanobacteria grow upward through the sediment, toward the light, forming a new living layer on top of the sediment layer. Another thin layer of sediment accumulates. Another layer of cyanobacteria grows upward, and so on year after year, layer by layer. The stromatolite grows upward a few tenths of a millimeter per year, a centimeter every few decades. The modern stromatolites of Shark Bay are rare. They exist here because the water is hypersaline, too salty for the invertebrates that would otherwise graze on the microbial mat and prevent the stromatolite from forming. In the modern ocean, cyanobacterial mats are kept in check by grazers. The stromatolite can only grow where grazers cannot survive. But in the Archean and Proterozoic world before complex grazers existed, the stromatolites were not rare. They were everywhere. Fossil stromatolites have been found in rocks from Western Australia, from Southern Africa, from Russia, from Canada, from dozens of other locations. Rocks that are 3.5 billion years old, 3 billion years old, 2.5 billion years old. The shallow margins of Archean and Proterozoic continents were covered in stromatolites. The seafloors of the Archean shallow seas were carpeted in microbial mats, and wherever conditions were right for their growth, the stromatolite columns rose from those mats. These stromatolites were built by ancient cyanobacteria, and the cyanobacteria were producing oxygen. Every stromatolite, every microbial mat, every square centimeter of cyanobacterial growth in the sunlit shallows of the ancient ocean was producing oxygen, tiny quantities per cell, per square centimeter, per day, but enormous in aggregate across the entire ocean surface, across millions of years. The stromatolites were patient. They had no concept of what they were doing. They were simply growing, simply photosynthesizing, simply producing oxygen, because that was what their metabolism produced. And across hundreds of millions of years of this patient, mindless, continuous oxygen production, they were changing the world. Cyanobacteria, the most dangerous organism that has ever lived. The cyanobacteria require a moment of genuine attention because they are perhaps the most consequential organisms in the history of life on this planet, and they are almost entirely unknown to most people. The name comes from the Greek for blue-green, kyanos, blue-green algae. They are sometimes called, though they are bacteria, not algae. They are prokaryote cells without nuclei, without mitochondria, without the complex internal structure of eukaryotic cells. By the standards of visible biology, they are utterly unremarkable. Microscopic rods and spheres and filaments, invisible to the naked eye, indistinguishable from 1,000 other types of bacteria without careful biochemical analysis. But what cyanobacteria do is extraordinary. They perform oxygenic photosynthesis, the same type of photosynthesis that every plant and alga on Earth performs, the photosynthesis that runs the biosphere. They split water molecules, H2O, using the energy of sunlight, extracting electrons and protons from the water, and release the remaining oxygen as a waste product. The electrons and protons are used to reduce carbon dioxide, building organic molecules, sugars, proteins, fats that the cell uses as structural material and energy storage. The split water molecules are key. Water is the electron donor, and when you split a water molecule, you get oxygen specifically. You get O2, molecular oxygen, the gas that now makes up 21% of the atmosphere. The cyanobacteria did not plan this. Oxygen was simply the leftovers, the exhaust, the thing left over when you strip the useful parts from a water molecule. Before the cyanobacteria evolved sometime between 3.0 and 2.7 billion years ago, based on the best current evidence, no organism on Earth could perform oxygenic photosynthesis. The anoxygenic photosynthesizers existed, as we have discussed, and they used sunlight for energy. But they used hydrogen sulfide or ferrous iron as their electron donors, not water. They produced sulfur or iron oxides as waste, not oxygen. The innovation that the cyanobacteria made using water as an electron donor was an extraordinarily difficult biochemical problem. Water is not a good electron donor. It is chemically stable, hard to oxidize, resistant to the process of giving up its electrons. To split a water molecule using sunlight requires a specific, sophisticated molecular machine. The oxygen evolving complex, a cluster of 4 manganese atoms and a calcium atom at the heart of the photosystem II complex, which is one of the most energetically demanding reactions in biology. Assembling this machine, evolving it from scratch, was one of the great achievements of evolution, a problem that evolution solved once, approximately 2.7 billion years ago, and has not needed to solve again since, because every photosynthetic organism on Earth today either descended from the cyanobacteria or acquired the ability from them. Once the cyanobacteria evolved, they had an essentially unlimited source of electrons. Water is everywhere in the ocean, where previous photosynthesizers were limited by the availability of hydrogen sulfide or ferrous iron, both of which occur in specific environments. The cyanobacteria could photosynthesize anywhere there was sunlight and water, which was everywhere at the ocean surface. The competitive advantage was enormous. The cyanobacteria could grow in environments where no other photosynthesizer could. They were not limited to the reducing, hydrogen sulfide-rich environments near volcanic vents and deep water upwellings. They could colonize the open ocean surface, the shallow shelves, the quiet lagoons, everywhere that sunlight penetrated the water. And they grew, and as they grew, they exhaled. The slow apocalypse begins. The oxygen produced by the first cyanobacteria did not immediately appear in the atmosphere. It could not. The atmosphere and the ocean were full of reducing substances that reacted immediately with any oxygen produced. Ferrous iron, methane, hydrogen sulfide, elemental sulfur, all of these sinks for oxygen were present in enormous quantities, and they consumed oxygen as fast as the cyanobacteria could produce it. This consuming phase lasted for hundreds of millions of years, for perhaps 300 million years, from roughly 2.7 billion years ago to roughly 2.4 billion years ago. The cyanobacteria were producing oxygen, and the oxygen was being immediately consumed by the reducing chemistry of the ancient ocean and atmosphere. The atmosphere remained anoxic. The ocean remained anoxic, but the sink was being depleted. The most visible evidence of this depleting sink is the banded iron formations, the BIFs. These are geological structures of extraordinary beauty and economic importance. Alternating layers of iron-rich and silica-rich rock, red and white, preserved in ancient sedimentary sequences on every continent. The iron in the red layers is ferric iron, oxidized iron, rust precipitated from the ocean water when the oxygen produced by the cyanobacteria met the ferrous iron dissolved in the water. The banded iron formations are a record of the oxygen sink being filled. Every layer of ferric iron represents another increment of the ocean's iron budget being oxidized, the dissolved ferrous iron being converted to insoluble ferric oxides and settling to the seafloor. The formations are layered because the rate of iron oxidation varied seasonally or cyclically, alternating between periods of more intense cyanobacterial activity and more oxidation, and periods of less activity and more iron dissolution from the seafloor. But the overall trend was clear. The iron was being consumed. The sink was shrinking. When the dissolved iron in the ocean had been largely consumed, when the BIF forming phase was ending, roughly 2.4 billion years ago, the oxygen had nowhere left to go. The sink was gone, or nearly gone. The oxygen produced by the cyanobacteria began to accumulate in the ocean, and then, slowly, it began to outgas into the atmosphere. The transition from essentially zero oxygen in the atmosphere to the first detectable, persistent oxygen levels is called the Great Oxidation Event, and it was not, by geological standards, instantaneous. It took perhaps 200 million years, from about 2.45 to 2.22 billion years ago, for atmospheric oxygen to rise from essentially nothing to perhaps 1 to 10% of current levels. But even 1% of current levels, roughly 0.2% of the atmosphere, was enough to fundamentally transform the chemistry of the world. And that transformation was the death of the old world, rusting the ocean. Before the oxygen reached the atmosphere, it destroyed the ocean, not quickly, not violently, but comprehensively. The rising oxygen concentrations in the ocean water, still very low by modern standards, but far above zero, began to oxidize the dissolved iron that had characterized the Archean Ocean for 2 billion years. The iron precipitated, the rust fell, and across millions of years, the deep purple-red color of the iron-rich ocean faded as the dissolved iron was removed from the water and deposited on the seafloor in the massive iron formations that we mine today. But the iron precipitation was not the only chemical transformation. The hydrogen sulfide produced constantly by the sulfate-reducing bacteria in the sediment and the deep water also reacted with the rising oxygen, being oxidized to sulfate. The methane in the atmosphere produced by the methanogens began to react with the oxygen in the lower atmosphere, being oxidized to carbon dioxide and water. The organic haze that had colored the Archean sky was slowly being burned away, as the methane that produced it was consumed by reaction with oxygen. This destruction of the methane was, as we will discuss, a catastrophe in its own right. But first, the chemistry of the ocean was fundamentally changing, the reducing chemistry that had prevailed for 2 billion years, giving way, gradually, to the oxidizing chemistry that characterizes the modern ocean. The organisms that had evolved in the reducing ocean, the iron reducers, the sulfate reducers, the fermenters, the hydrogen consumers, found their world shrinking, not quickly. Oxygen, at the concentrations it was reaching in the early Proterozoic ocean, was not immediately lethal to all anaerobes, but it was toxic to many of them. The enzymes of the anaerobic metabolism, the specific molecular machines evolved to function in reducing conditions, were damaged by oxygen. The oxygen attacked them, oxidized the iron-sulfur clusters at their active sites, disrupted their function. For organisms that had evolved in the complete absence of oxygen, the rising concentrations were an existential threat. Not the threat of a sudden catastrophe, not an asteroid or a volcanic eruption, but the slow, relentless, chemically inevitable threat of a world that was becoming hostile to their fundamental biology. They could retreat. Some of them did, moving to the deep sediments, to the reducing environments deep below the seafloor, to the isolated anoxic basins where oxygen had not yet penetrated. Some developed strategies for dealing with low levels of oxygen, the beginnings of the antioxidant biochemistry that would eventually become the sophisticated reactive oxygen species management of modern aerobic cells. Some became the ancestors of the organisms that would eventually embrace oxygen and run their metabolism on it, but many died. The Great Oxidation Event, Earth's first mass extinction. The Great Oxidation Event is not discussed much outside of specialist scientific circles. It has no popular name beyond its scientific designation. It has no museum exhibit. It has no science fiction films. It does not appear in the cultural consciousness the way the extinction of the dinosaurs appears, or the Ice Age, or even the Permian extinction. And yet it was, by any measure that matters, the largest and most comprehensive extinction event in Earth's history. The Permian Triassic extinction, the Great Dying, killed 90% of marine species and 70% of land species. The End Cretaceous extinction killed 75% of all species. The Great Oxidation Event killed or drove into refugia so marginal and so restricted that extinction at the population level was universal, a fraction of life that is almost impossible to quantify because we do not have a complete census of the organisms that existed before the oxidation and a complete census of the organisms that existed after. What we know is this: the anaerobic world that had existed for 2 billion years, the world of the methanogens and the iron reducers and the sulfate reducers and the anoxygenic photosynthesizers, was destroyed as a globally dominant ecosystem. These organisms did not all go extinct. They survive today in their refugia, in the deep sediments and the anoxic water columns of stratified lakes and the digestive systems of animals. But they were driven from the surface ocean. They were driven from the atmosphere. They were driven from the shallow water, the sunlit zones, the productive upper ocean. The ecological space they had occupied for 2 billion years was taken from them permanently by the oxygen. The scale of this displacement is staggering. The anaerobic metabolism community of the Archean Ocean was not a handful of species in a few specialized environments. It was the entire living world, billions of different metabolic strategies, billions of years of evolution, the full diversity of microbial life in an anoxic setting, all of it pushed to the margins, most of it gone from the dominant ecological stage, never to return. The closest analogy in the visible world, the world of organisms large enough to leave fossils that allow us to count extinctions, would be if a catastrophe killed not just specific animal and plant species, but the entire concept of multicellular life, not just certain dinosaurs or certain fish, but every eukaryote, every multicellular organism, every complex life form. That is the scale of what the Great Oxidation Event did to the anaerobic world. It did not just change the species composition of the living world. It ended a 2 billion year era of anaerobic dominance and began something entirely new. The methane crisis and the first snowball. The destruction of the anaerobic world was not the only consequence of the Great Oxidation Event. The rising oxygen had another, more immediate, more dramatic effect. It destroyed the methane greenhouse. The methane in the Archean atmosphere, produced by the methanogens, maintained at high concentrations by the reducing chemistry of the ocean and atmosphere, was the primary greenhouse gas, keeping the Earth warm under the dim, young sun. Methane is a powerful greenhouse gas, approximately 30 times more potent than carbon dioxide over a century-long timescale. In the Archean atmosphere, where methane concentrations may have been hundreds to thousands of times higher than today, the methane greenhouse was the difference between a liquid water world and a frozen snowball. When oxygen began accumulating in the atmosphere, it reacted with the methane. Oxygen and methane in the atmosphere undergo a slow chemical reaction, not combustion, not in the thin, cold atmosphere, but a photochemical reaction catalyzed by ultraviolet radiation. The oxygen destroys the methane, converting it to carbon dioxide and water. The methane concentration falls. The greenhouse weakens. The planet cools. This cooling, triggered by the same oxygen that was simultaneously poisoning the anaerobic biosphere, pushed the Earth into the most severe glaciation in its history, approximately 2.4 to 2.1 billion years ago. During the Huronian glaciation, the Earth may have experienced the first snowball Earth event, a glaciation so extreme that the ice sheets extended to the equator, covering the entire surface of the planet in ice. A snowball Earth is the most extreme climate state the planet can achieve, with the surface entirely covered in white ice. The albedo, the fraction of sunlight reflected back to space, is enormous. Most of the sunlight arriving at the Earth is simply reflected away, and the cooling feedback becomes self-reinforcing. The ice spreads, the albedo increases, more cooling occurs, more ice spreads. The only way to escape is through volcanism, the slow, geological timescale accumulation of carbon dioxide from volcanic outgassing, until the greenhouse effect becomes strong enough to begin melting the ice. The Huronian snowball, if it was truly a snowball, and not simply a very severe glaciation, lasted perhaps 300 million years. For 300 million years, the Earth may have been entirely frozen, the ocean covered by hundreds of meters of ice, the land buried under ice sheets kilometers thick, the only liquid water in the world at the hot vents on the seafloor, where volcanic heat maintained small pockets of liquid even under the ice. Life survived, as it always survives, in those seafloor hot springs, in the tiny pockets of liquid water beneath the ice, in the refugia of the deep sediment where liquid water persisted. Life survived. The methanogens, driven from the open ocean but not from the deep hydrothermal environments, continued their slow metabolism in the dark. The iron reducers continued. The sulfate reducers continued. Life held on, as it always holds on, at the margins, in the cracks, in the places too extreme for even the oxygen to reach. The snowball ended when volcanic CO2 accumulated enough to melt the ice. The melt was sudden once the ice began to thin and the albedo began to decrease. The positive feedback ran in reverse, and the planet warmed rapidly from a frozen snowball to a briefly hothouse world, as the accumulated volcanic CO2 drove temperatures far above modern levels. The aftermath of the snowball was a world of extreme temperature swings, of complicated ocean chemistry, of disrupted biological communities struggling to re-establish themselves in a drastically altered environment. This was the world that emerged from the Great Oxidation Event, transformed, impoverished, cold, and strange. The ancient anaerobic Eden was gone. In its place was a world that, for the first time, had oxygen in its atmosphere and had no idea what to do with it yet. The survivors, life in the margins, the anaerobic organisms that survived the Great Oxidation Event, the ones that persist today in their marginal, restricted environments, tell us what was left after the apocalypse. They are there today, if you know where to look. Pull a handful of mud from the bottom of a pond and bring it to the surface. As you stir it, it will release a faint smell of hydrogen sulfide and methane, the exhalations of the sulfate reducers and the methanogens living in the anoxic sediment, a millimeter or two below the surface, breathing sulfate and carbon dioxide in conditions as reducing as anything in the Archean Ocean. These organisms are not relics or fossils. They are alive, active, doing exactly what their ancestors have been doing for 3 billion years. They survived the Great Oxidation Event by retreating into the sediment, into the deep and reducing conditions that the oxygen could not reach, and they have been there ever since. The methanogens are in every wetland, every rice paddy, every ruminant's gut. The methane that cattle produce, the subject of much discussion in the context of climate change, is produced by methanogens in the rumen, doing exactly what methanogens have always done, consuming hydrogen and carbon dioxide, producing methane. The methane in the atmosphere today, not just from cattle but from wetlands, from the Arctic permafrost, from rice paddies, is largely biological in origin, the exhaled waste of methanogens that have been performing this metabolism for 3 billion years. They are here. They survived. The iron-reducing bacteria are in the sediment of rivers and lakes, reducing iron minerals in conditions too reducing for oxygen to persist. The sulfate-reducing bacteria are in the anaerobic zones of the ocean, the Black Sea, the deep waters of some fjords, the sediment of continental shelves, maintaining the sulfur cycle that has been running since before oxygen appeared. These organisms are the living memory of the pre-oxygen world. They carry in their DNA the oldest biochemical strategies that life has discovered, strategies that were already ancient when the cyanobacteria first appeared. They survived the apocalypse by going underground, literally and figuratively, and waiting in the margins, while the new, oxygen-rich world established itself around them. And in those margins, In the billions of years since the Great Oxidation event, they have continued to play essential roles in the Earth's biogeochemical cycles. The sulfur cycle, the iron cycle, the methane cycle, all of these still run in the anoxic niches of the modern world, driven by the organisms that predate oxygen. The modern Earth is not a purely aerobic world. It is a world with two metabolic layers: the aerobic surface, with its oxygen and its complex life, and the anaerobic deep, with its ancient chemistry and its ancient organisms. Both layers are essential. The modern biogeochemical cycles cannot run without both. The boring billion, a world suspended. After the Great Oxidation event, after the Huronian snowball, after the collapse of the ancient anaerobic world, the Earth entered a period that geologists have named, with unusual candor, the boring billion. From approximately 1.8 billion to 0.8 billion years ago, a stretch of time that is longer than the entire history of complex animal life. Longer than the time since the Cambrian explosion, the Earth maintained a state of relative stagnation. The oxygen levels, after rising during the Great Oxidation event, did not continue to rise. They stabilized at low levels, perhaps 1 to 10% of the present atmospheric level, and stayed there for approximately 1 billion years. The ocean remained mostly anoxic below the surface layers. The deep ocean was still reducing, still sulfidic, still hostile to aerobic life. The shallow marine environments were oxygenated, but the bulk of the ocean volume was not. In this environment, complex life did not evolve. The eukaryote cells with nuclei, the lineage that would eventually produce all complex organisms, had appeared by approximately 2 billion years ago, perhaps earlier. But for 1 billion years, they remained single-celled. The microbial world remained the dominant world. Stromatolites continued to build their layered columns in the shallow seas. The cyanobacteria continued to produce oxygen. The anaerobes continued to occupy the deep water and the sediment, and nothing very new happened. Why? The answer appears to be oxygen specifically, the insufficiency of it. Complex multicellular organisms require relatively high concentrations of dissolved oxygen to function. The oxygen demands of a body made of millions of cells, with its requirement for diffusion of oxygen to every cell, with the metabolic needs of differentiated tissues performing different functions, are much higher than the oxygen demands of a single-celled organism. A single-celled eukaryote can function at the low oxygen concentrations of the Proterozoic ocean. A complex multicellular organism cannot. The boring billion was a waiting period. Life was waiting for the oxygen to rise. The eukaryotes had the machinery for complexity, the nucleus, the mitochondria, the endomembrane system, the cytoskeleton, but they could not deploy it fully until the oxygen concentration rose to the level where complex multicellular metabolism was energetically feasible, and the oxygen was not rising because the nutrient cycles of the Proterozoic ocean were configured in a way that limited the productivity of the cyanobacteria and therefore limited the rate of oxygen production. The boring billion ended when the Neoproterozoic snowball events, the Sturtian and Marinoan glaciations, approximately 720 and 635 million years ago, disrupted the stable but unproductive configuration of the ocean. The snowball events mixed the ocean, redistributing nutrients, oxygenating deep water, disrupting the stratification that had kept the deep ocean anoxic for 1 billion years. When the ice melted, the ocean that emerged was more oxygenated, more nutrient-rich, more productive than the Proterozoic ocean had been. The stage was set for something new. Why complexity required poison? Here is the paradox at the heart of this story, and it is worth sitting with for a moment before we move on. Complex life, the kind of life that produces fish and forests and dinosaurs and mammals and human beings, required oxygen, not in the way that we sometimes say things require things, meaning it would be convenient or beneficial, required in the hard, thermodynamic sense. Without oxygen, aerobic respiration is impossible. Without aerobic respiration, the energy yield from metabolism is so low that a complex multicellular organism cannot sustain itself. And yet the oxygen that complex life requires was, for the organisms that existed before it, a poison. The great oxidation event, the arrival of the oxygen, destroyed the ancient anaerobic world. It killed or marginalized the creatures that had existed for 2 billion years. It triggered a snowball Earth. It was, by any reasonable measure, a catastrophe. The paradox is, the catastrophe was necessary, not in a teleological sense, not because the universe had a plan, or because evolution was heading somewhere predetermined, but in the mechanical, physical sense. The conditions that allowed the evolution of complex life were only established by the destruction of the conditions that had preceded them. The oxygen that killed the Archean world was the same oxygen that powered the Cambrian world. The poison was the gift. The apocalypse was the prerequisite. This is one of the most profound lessons in the history of life on Earth. The progress of life is not a story of continuous improvement, from the simple to the complex, from the inferior to the superior. It is a story of transformations, catastrophic transformations, in many cases that destroy existing worlds and create new ones. The organisms of the old world are not inferior to the organisms of the new world. They are different, adapted to different conditions. When the conditions change, they are displaced, and from that displacement, that emptying, that reshaping, something new becomes possible. The oxygen was both the worst thing that ever happened to the living world and the necessary condition for everything that came after. To mourn the loss of the anaerobic Eden is appropriate. It was a world of extraordinary richness and complexity, a world that lasted 2 billion years, a world that we can glimpse only in the most ancient rocks and in the marginal survivors that still practice the ancient chemistries in the sediments of ponds and wetlands. But to celebrate the oxygen is also appropriate, because without it, there is no you, there is no thought, there is no one to mourn or celebrate anything. The oxygen that is filling your lungs right now, the poison that killed the Archean world, is the reason that you exist to think about it. The mitochondria revolution. To understand why oxygen was so transformative for the evolution of complex life, we need to understand the mitochondria, the organelles within your cells that use oxygen to produce energy. The mitochondria are not originally yours. They are not originally anyone's. They were, approximately 2 billion years ago, free-living bacteria, specifically alpha proteobacteria, a group of aerobic bacteria that had evolved the machinery for aerobic respiration and were using oxygen to run their metabolism with the efficiency that only aerobic respiration allows. They were small, free-living organisms, swimming in the early oxygenated ocean, running their aerobic metabolism, doubling every few hours. At some point, the exact circumstances are unknown, but the event was singular and transformative. One of these aerobic bacteria was taken up by a larger cell, not digested, not killed, taken up and allowed to persist inside the larger cell. still running its aerobic metabolism, still producing ATP from oxygen, and the larger cell, instead of destroying the intruder, kept it. The aerobic bacteria inside the cell was producing ATP in quantities far beyond what the cell could produce on its own. It was the most productive thing that had ever happened to the larger cell. This event, the endosymbiotic acquisition of the mitochondria, is one of the 2 or 3 most consequential events in the history of life on Earth. The cell that acquired the mitochondria now had access to aerobic respiration to the 18 times more productive energy metabolism that oxygen enables. It could support larger body size, more complex cellular machinery, more energetically expensive processes. It could afford to be complex. The cell with mitochondria is the eukaryotic cell, your cell, my cell, every cell in every complex organism alive today. Every animal, every plant, every fungus, every protist, all cells with nuclei contain mitochondria, or the remnants of mitochondria, because they are all descended from that single, ancient endosymbiotic event. The mitochondria are your oldest ancestors, oldest prisoners. They have been living inside eukaryotic cells for 2 billion years, running their aerobic metabolism, producing the ATP that powers the complexity of your existence. Without the mitochondria, no complex life. Without oxygen, no mitochondria, or rather, mitochondria that cannot do what mitochondria do, cannot run the oxygen-consuming reactions that produce the ATP. Without the great oxidation event, no oxygen in sufficient concentrations to power aerobic respiration at the cellular level. Without the poison, no mitochondria, no eukaryotes, no complex life. The chain of necessity runs from the apocalypse to you, through the mitochondria, through the endosymbiotic event, through the rising oxygen of the Proterozoic ocean. You are made of the consequences of a catastrophe, oxygen and the edge of fire. There is one more consequence of oxygen that deserves attention before we follow the story forward. Fire. Fire is an oxidation reaction, a rapid, exothermic, self-sustaining oxidation reaction in which organic matter, wood, grass, leaf litter, reacts with atmospheric oxygen to produce carbon dioxide, water, and heat. Fire requires 3 things: fuel, oxygen, and ignition. On the pre-oxygen Earth, 2 of these were available. There was organic matter, the bodies of microorganisms, the products of their metabolism, and there was ignition, lightning, volcanic activity, but there was no oxygen. Fire was impossible. When oxygen accumulated in the atmosphere, fire became possible for the first time. The Devonian and Carboniferous periods, when forests were established and oxygen levels rose to perhaps 35% of the atmosphere, were also the first great ages of fire. Charcoal fossil charred wood appears in the rock record for the first time in the Devonian, direct evidence of wildfires burning through the first forests. The Carboniferous is extensively documented to have been an age of frequent, large wildfires, driven by the extraordinarily high oxygen levels and the abundant fuel of the vast coal swamp forests. Fire is one of the most powerful ecological forces on Earth. It clears vegetation, cycles nutrients, creates new habitat, selects for fire-resistant species and fire-dependent species. Many modern ecosystems, the savannas of Africa, the pine forests of North America, the heathlands of Europe, are maintained by fire. Many plants have evolved traits specifically for surviving or exploiting fires. The serotinous cones of certain pines, that only open and release seeds in the heat of a fire, the thick bark of giant sequoias that protects them from all but the most intense fires, the deep root systems of grasses that allow rapid regrowth after fires consume the above-ground parts. None of this would exist without oxygen. Fire is oxygen's gift or oxygen's curse, depending on your perspective. In the world without oxygen, there is no fire. The forests of a world without oxygen, if there were forests, which without oxygen there could not be, would be completely immune to fire. The ecology of a world without oxygen is an ecology without fire, which is an ecology profoundly different from any terrestrial ecology we know. The Cambrian gift. The oxygen levels continued to rise through the Neoproterozoic, and by the time the Ediacaran period began approximately 635 million years ago, the ocean was sufficiently oxygenated to support the first complex, multicellular animals. The Ediacaran organisms Dickinsonia and Charnia and Tribrachidium and their extraordinary kin were the first multicellular animals with bodies visible to the naked eye. They were simple, no hard parts, no complex organs, no predators, no prey in the conventional sense, but they were multicellular, which means their cells were specialized, different cells doing different jobs, which means they required the energy production that only aerobic respiration could provide at the necessary scale. They were possible because the oxygen was, by the Ediacaran, high enough. And then came the Cambrian explosion 541 million years ago, the sudden and extraordinary diversification of animal body plans that produced essentially all the major animal lineages that have existed on Earth. The arthropods, the mollusks, the echinoderms, the chordates, the ancestors of every invertebrate and every vertebrate that has ever lived, all appear in the fossil record of the Cambrian within a few tens of millions of years. The Cambrian explosion was powered by oxygen, not solely ecological factors, the development of predation, the available of hard part mineralization strategies, the global geography of the time all contributed, but the energy budget made available by aerobic respiration in a sufficiently oxygenated ocean was a fundamental prerequisite. The Cambrian animals were large by the standards of what had come before. Active, fast-moving, metabolically expensive organisms, they could only exist in an ocean with enough dissolved oxygen to run their energetic lives. Without the great oxidation event, without the poison, the Cambrian explosion does not happen. The ocean in which it occurred is the direct descendant of the great oxidation event, modified by 1 billion years of continuing oxygen accumulation and ocean chemistry evolution. The animals of the Cambrian breathed the legacy of the cyanobacteria's ancient, mindless, world-destroying exhalation. The Devonian and the first breathing forests. 400 million years ago, the Devonian period. Oxygen levels in the atmosphere are rising, perhaps reaching 15% by the end of the Devonian, compared to 21% today. On land, something extraordinary is happening. The first forests are growing. The first forests were not forests in the sense we know. They were dominated by plants without leaves, simple, upright, green stems and branches, catching sunlight along their entire above-ground surface. The trees of the early Devonian Cooksonia, Aglophyton, the progenitors of all later forest trees, were small and structurally simple, but they were photosynthesizing and they were making oxygen. The expansion of land plants in the Devonian had a dramatic effect on atmospheric oxygen. Land plants are prolific oxygen producers. They take carbon dioxide from the air, fix it into organic matter using sunlight, and release oxygen. As the forests spread across the Devonian continent slowly, across millions of years, expanding from the coastal wetlands into the drier interiors, they were producing oxygen on an unprecedented scale. By the late Devonian, the vast lycopsid forests and seed fern forests were significant contributors to the atmospheric oxygen budget, but the forests of the Devonian and Carboniferous also did something else. They pulled carbon dioxide out of the atmosphere and stored it in their wood. Lignin, the structural polymer that makes wood hard and resistant to decomposition, is rich in organic carbon. In the Devonian and Carboniferous, when the organisms capable of efficiently decomposing lignin had not yet evolved, the lignin-rich wood of the early forests accumulated without fully decomposing. It was buried. It became coal. Carbon that had been in the atmosphere as CO2 was now locked in the ground as coal, and the oxygen that had been produced when that carbon was fixed remained in the atmosphere. This is the origin of the coal that powered the Industrial Revolution. The coal of the Carboniferous, the coal seams of Pennsylvania and Wales and the Ruhr Valley, is the fossilized wood of the Carboniferous forests, and the oxygen that our civilization runs on today, the oxygen that is 21% of the modern atmosphere, is, in part, the oxygen that was produced when those trees grew and that remained in the atmosphere when their wood was buried without being fully oxidized. The air you breathe is, in part, the legacy of a Carboniferous tree. The Carboniferous atmosphere, too much of a good thing. The Carboniferous period, 359 to 290, 9 million years ago, is famous for two things: its giant insects and its vast coal swamp forests. Both are consequences of the same phenomenon: abnormally high atmospheric oxygen. By the mid-Carboniferous, atmospheric oxygen levels had risen to approximately 30 to 35%, significantly above the modern 21%. The reasons were, as discussed, the burial of vast quantities of organic carbon in the lignin-rich coal swamps, combined with the continued expansion of land plant cover and its associated photosynthetic oxygen production. The oxygen levels of the Carboniferous were the highest the Earth has experienced since. The effects were dramatic. Insects, which breathe through passive diffusion along tubes called tracheae, are limited in body size by the efficiency of this diffusion system. In normal air, the maximum efficient size for a tracheal respiratory system is roughly the size of a large cockroach. But in the high oxygen atmosphere of the Carboniferous, diffusion was faster and more efficient, and insects could grow much larger while still getting adequate oxygen to all their tissues. The result was the giant insects of the Carboniferous. Meganeura, a dragonfly with a wingspan of 65 to 70 centimeters. Arthropleura, a millipede reaching 2.5 meters in length. Pulmonoscorpius, a scorpion perhaps 70 centimeters long. These giants were the product of too much oxygen of the atmosphere, pushing past the optimum and into a range that enabled body sizes that have never been replicated before or since. The Carboniferous was the age of enormous insects because the air was extraordinarily rich in the gas that powers life. But the high oxygen also meant high fire risk. Modern high oxygen environments where oxygen exceeds about 25% are substantially more flammable than the modern atmosphere. Lightning ignition of dry vegetation is more likely. Fires burn hotter, faster, and are harder to extinguish. The Carboniferous fossil record contains abundant charcoal fossil burned wood, evidence of frequent, intense wildfires sweeping through the coal swamp forests. The forests that were building the coal and producing the oxygen were also burning, cycling through growth and fire in rhythms driven by the climate and the oxygen concentration. The Carboniferous atmosphere was extreme. Too much oxygen, too much fire, too much insect. The world had overshot the optimal, driven by the extraordinary carbon burial efficiency of the first wood-producing forests. Eventually, the oxygen levels came down. As the oxygen sinks, respiration, fire, the oxidation of buried organic matter by erosion caught up with the sources. By the Permian, oxygen levels had declined toward more moderate values, and the giant insects shrank back to the sizes we know today. The modern oxygen economy. The oxygen level in the modern atmosphere, 21%, is not an accident. It is the result of a long-term balance between the processes that produce oxygen, photosynthesis, by land, plants, and marine algae, and the processes that consume it, respiration by animals and microbes, the oxidation of organic carbon and other reducing materials during weathering, and volcanic outgassing of reducing gases. This balance has been maintained, with fluctuations, for hundreds of millions of years. The specific value of 21% appears to be, in a rough sense, optimal for complex terrestrial life, high enough to support the metabolic needs of large, active animals, low enough that wildfires, though present, do not consume every terrestrial ecosystem. Above about 25% oxygen, any ignition source would set fire to even wet vegetation. The modern atmosphere is in a zone where fire is possible and important as an ecological force, but not so common that it prevents the accumulation of vegetation and soil. The photosynthetic organisms that maintain this oxygen level, the phytoplankton of the ocean and the plants of the land surface, are running the same basic reaction that the cyanobacteria discovered 2.7 billion years ago. Every plant on Earth is, in a metabolic sense, a cyanobacterium, or rather, the descendant of a eukaryotic cell that engulfed a cyanobacterium and never let it go. The chloroplasts in every plant cell, the green, light-harvesting organelles that perform photosynthesis, are the direct descendants of ancient cyanobacteria, acquired in a second great endosymbiotic event that preceded the evolution of all photosynthetic eukaryotes. The cyanobacteria are still in your cells if you are a plant, in the cells of the trees outside your window, in the cells of every blade of grass. They have been working for 2.7 billion years, splitting water molecules, releasing oxygen, maintaining the atmosphere that makes you possible. The most dangerous organism in the history of life is also the most generous. What the world would be without it? We have been circling this question throughout the night, approaching it from different angles, gathering the pieces of the picture. Now let us look at it directly. If the cyanobacteria had never evolved, if the great oxidation event had never occurred, if the oxygen had never appeared in the atmosphere, not at 2.4 billion years ago, not ever, what would the Earth be tonight? The first and most obvious answer is a world of microbes. Without oxygen, aerobic respiration is impossible. Without aerobic respiration, the energy budget available to any multicellular organism is approximately 18 times smaller than in our world. The eukaryotic cell might have evolved the endosymbiotic event might have occurred, though without the selective pressure of an oxygenated environment, the timing and the outcome might be different. But the eukaryotic cell, without mitochondria, running aerobic respiration, is a much less powerful cell. A cell that can support only the most limited forms of multicellularity. The boring billion in the world without oxygen would not have ended. It would have continued. The single-celled eukaryotes, limited by the low energy yield of anaerobic metabolism, would not have been able to sustain the metabolic investment required for complex, multicellular bodies. The Ediacaran organisms would not have appeared. The Cambrian explosion would not have occurred. There would be no animals, no plants, no fungi, no visible life, just the ocean of microbes, the ancient, diverse, chemically complex microbial world of the Archean and Proterozoic, extending, unchanged, to the present day. The ocean tonight, without oxygen, would be a purple-red ocean, colored by dissolved iron. The atmosphere would be brownish-orange, colored by methane haze. The land surface would be bare rock and mineral deposits, with thin films of microbial mat on the wettest surfaces, but nothing that could be called vegetation. There would be no forests, no grasslands, no soils in the modern sense. There would be no animals, no insects, no fish, no reptiles, no mammals, no birds, no footprints anywhere, no tracks, no burrows, no bones. The sounds of the world without oxygen, the wind, unimpeded by any vegetation that might buffer it, moving over bare rock and sand with a hollow, empty sound. The waves of the ocean, reaching a shoreline with no tide pools, no kelp, no barnacles, no intertidal life. The distant sound of volcanoes still active, as they always are, adding carbon dioxide and sulfur dioxide and hydrogen sulfide to the atmosphere. No bird song, no insect humming, no rustle of leaves, no sound of any animal breathing. The sky would be spectacular, the methane haze creating extraordinary sunsets in orange and red and purple, more vivid than any modern sunset. And at night the stars would be magnificent, unmarred by any light pollution, visible through the relatively clear high atmosphere. But there would be no one to see the stars, no one to find them beautiful, no one to name them, or tell stories about them, or navigate by them. The world without oxygen would be a world of extraordinary chemical complexity and biological diversity at the microbial scale. It would be a world of ancient, exquisitely refined metabolisms, of chemical gradients and electron transfers, and the slow cycling of elements through the ocean and the atmosphere. It would be a world that has been running its chemistry for 4 billion years, with no interruption. It would be, in its microbial scale, a world of remarkable richness, but it would be, by every human measure, empty. The anaerobic Earth today. The anaerobic Earth of the Archean does not exist only in the past. It exists today, hidden beneath the surface of the aerobic world, in every anoxic niche that the aerobic world has failed to fully colonize. Dive to the bottom of the Black Sea, below 150 meters. The water column is completely anoxic. The deepest water of the Black Sea has been anoxic for thousands of years, since the basin became stratified and isolated from the oxygen-rich surface water. In this anoxic zone, the water is rich in hydrogen sulfide, colored faintly by dissolved iron, alive with sulfate-reducing bacteria and methanogens and other anaerobes. It is a small piece of the Archean Ocean, preserved in the modern world, by the specific geography of the Black Sea Basin. Fly over the Amazon basin on a clear day and look at the rivers. The dark, tannin-stained waters of the Rio Negro, the Black River, carry organic acids from the flooded forests of the Amazon, and in the still, deep pools along its course, the bottom water is anoxic, occupied by the same sulfate reducers and methanogens that lived in the Archean Ocean. Below the surface organic-rich layer of tropical soils, the reducing conditions support iron-reducing bacteria that process the iron minerals of the laterite soils. In every wetland, every marsh, every waterlogged soil, the anaerobic organisms are present, maintaining their ancient chemistries in the reducing conditions that persist wherever organic matter accumulates faster than oxygen can decompose it. They are the surviving citizens of the world that oxygen destroyed, present, active, essential to the modern biogeochemical cycles, but confined to the margins of a world that was taken from them 2.4 billion years ago. The strange organisms that still live without it. Some of the most extraordinary organisms on Earth are the ones that have never adapted to oxygen at all. The strict anaerobes, the organisms for which oxygen is still, after all this time, a poison. Clostridium. This bacterial genus includes some of the most medically significant organisms known. Clostridium botulinum, which produces botulinum toxin, the most toxic biological substance known. Clostridium tetani, which produces the toxin responsible for tetanus. Clostridium difficile, the scourge of hospital-acquired infections. All of these are strict anaerobes. Oxygen kills them. They exist in anoxic environments, soil, sediment, the anoxic conditions inside a wound. They are the most visible reminder that the anaerobic world is not gone, that it persists in the margins, and that it can be dangerous even in the oxygen-rich modern world. The thermophilic archaea of hydrothermal vents are perhaps the most dramatic anaerobic survivors. Pyrococcus furiosus, the furious fireball, is an archaeon found in hydrothermal vents near Volcano, Italy, that grows at temperatures up to 103 degrees Celsius and is completely unable to tolerate oxygen. It uses sulfur as a terminal electron acceptor, producing hydrogen sulfide as a waste product, the same metabolism that has been running in the anaerobic deep since the Archean. It is completely untouched by the oxygen revolution that transformed the surface world. Its world has not changed in 3 billion years. The foraminiferal cysts, found in the anoxic sediment of the Norwegian fjords, single-celled eukaryotes that have evolved mitochondria-like organelles that function without oxygen, using sulfate as an electron acceptor, represent one of the most recent evolutionary experiments in anaerobic eukaryotic life. These organisms, discovered in the early 2000s, demonstrate that the transition back to anaerobic metabolism is possible even from within the eukaryotic lineage. They are the eukaryotic world's tentative reconnection with its anaerobic past. And deep in the subsurface, in the deep continental crust, in the porous rock kilometers below the surface, where the water circulates on timescales of millions of years and has never been in contact with the atmosphere, there are microbial communities, so isolated, so ancient in their lineages, so removed from the oxygen world above them, that they might as well be Archean organisms. Some of these subsurface communities are sustained entirely by the chemical energy of the rock, by the reaction of water with iron minerals, producing hydrogen, which the hydrogen-consuming archaea use as fuel. They have not seen sunlight, in any evolutionary sense since before the Great Oxidation event. They are the most ancient survivors, the paradox at the center of everything. We have arrived at the place we were always heading, the paradox that makes this story worth telling. You are breathing oxygen right now, the gas that is powering your cells, that is running your metabolism, that is giving you the energy to hear this story and to think about it. This gas was once the most destructive substance in the history of life on Earth. It killed the world that preceded ours. It triggered the largest extinction event in the history of life, the first and most total of all the world endings that life has experienced. It poisoned the ancient ocean. It destroyed the methane greenhouse and plunged the world into a snowball. It was catastrophe. And yet, without it, you are not here. Without the Great Oxidation event, no complex life. Without complex life, no fish, no forests, no dinosaurs, no mammals. Without mammals, no primates. Without primates, no humans. Without humans, no one to lie in the dark tonight and breathe and think and wonder. The thing that almost ended everything is the thing that made everything possible. This is not a comfortable paradox. It does not resolve into a simple lesson or a clean moral. The organisms of the anaerobic world did not deserve to be destroyed. They were not inferior. They were not primitive. They were not an early draft that needed to be discarded for the final version to appear. They were complete in themselves. The world they inhabited was complete, self-sustaining, ancient, complex. Their destruction was a genuine loss, a 2-billion-year-old loss, and the fact that the destruction was the prerequisite for our existence does not make it less of a loss. But the paradox is also a kind of wonder, that the most destructive event in the history of life was simultaneously the most generative, that the poison was the gift, that the apocalypse was the foundation, that the catastrophe that ended the world the methanogens had built was the same event that built the world you were born into. Every breath you take is the product of that ancient catastrophe. Every thought in your head is powered by the oxygen that the cyanobacteria first exhaled 2.7 billion years ago. The mitochondria in your cells are the descendants of the aerobic bacteria that evolved in the first oxygenated ocean. Your existence is, from one angle, a story of extraordinary good fortune, the good fortune of being alive in the world that the poison made, rather than the world the poison destroyed, a night on the pre-oxygen Earth. Let us spend a moment there, one last moment, in the world that was. It is 3 billion years ago. The sky is orange-brown, the sun setting toward the horizon with a bloom of color unlike anything in the modern sky. The methane haze scattering the light in different frequencies, producing sunsets of amber and rust, and the deep purple red of an infrared photograph. There are no clouds in the modern sense the water cycle is operating, but the absence of cloud condensation nuclei from biological particles means the clouds form and move differently. The sky is vivid and alien and beautiful. The ocean stretches in every direction, colored by dissolved iron to a reddish brown, not the uniform redness of a rusted surface, but the variable, shifting color of a chemical mixture, darker where the iron concentration is highest, lighter where the surface has been depleted. The surface is still. There are waves, driven by the wind and by the closer, larger moon of 3 billion years ago. The moon was closer then, and tidal forces were stronger, but the surface lacks the glitter of a modern ocean because there is no oxygen in the atmosphere to produce the oxidized sea foam that whitens modern waves. The shore is bare rock, no sand. Exactly, sand is mostly the product of biological erosion, of organisms that grind shells and bones and reef material into fine particles. The Archean shore is coarse mineral grains, the products of purely physical and chemical weathering of rock. No beach, no dunes, no plants, no animals, just rock meeting water in the late orange light. In the shallows, rising from the sea floor in dome-like columns, the stromatolites are visible, the same structures that exist in Shark Bay today, but here covering vast areas of the shallow shelf, growing undisturbed by any grazer, coloring the shallow water with their microbial mats. Seen from the surface, in the orange light, they look like small islands. Seen from below, looking up through the water toward the sunset, they are dark columns against the glowing surface, the water between them murky with microbial particles. No sound of insects, no bird calls, no sound of any breathing animal, only the waves on the rock, the wind across the bare hills, the occasional distant volcanic rumble, the water. The world is not silent, it is full of physical sound, the deep sound of a living planet doing its geological work. But the biological silence is complete. No organism here makes a sound that can be heard above the water. And below the surface, below the orange reflecting shallows, in the deeper water where the light fades quickly to darkness, the chemistry of the Archean Ocean is at work. The methanogens are producing methane. The sulfate reducers are producing hydrogen sulfide. The iron oxidizers are processing iron at the chemical gradients. Billions of invisible organisms running their ancient metabolisms, cycling the elements of the ocean, maintaining the chemical world that they have built and that maintains them in return. It is a complete world, a whole world, a world that runs without you, without anything like you, without anything that breathes in the way you breathe. A world that has been complete for 1 billion years and will remain complete for another billion until the cyanobacteria change it forever. Look at it, the orange sky, the red-brown sea, the bare rock shore, the silent biological richness of the invisible world beneath the water. This is the world that oxygen killed. This is the world that you replaced. Epilogue. The first breath. Take another breath. A slow one. Feel the chest expand, the rib cage open, the air fill the space of the lungs. You are breathing oxygen, 21% of the air, the legacy of 2.7 billion years of cyanobacterial exhalation, of the great oxidation event, of the rusting of the ancient ocean, of the Huronian snowball, of the billion patient years of the Proterozoic, of the Cambrian explosion, of the Devonian forests, of the Carboniferous coal swamps, of all the millions of years of photosynthesis and burial and atmospheric accumulation that built the air you are breathing right now. This breath is the end of a very long story, a story that begins with a small, single-celled organism doing something that had never been done before, splitting a water molecule with sunlight, exhaling a gas that would change the world, a story that moves through catastrophe and extinction and snowball and recovery and the slow, patient accumulation of complexity in an increasingly oxygenated ocean, a story that passes through the Cambrian explosion and the Devonian forests and the Carboniferous giants and the Permian extinction and the rise of the dinosaurs and the asteroid and the rise of the mammals and all the millions of years of evolution that led from the first aerobic bacterium to the primate lying in the dark tonight, breathing. The first breath was a catastrophe. The cyanobacteria's first exhaled oxygen molecule, The very first molecule of O2 to enter the ancient atmosphere was the first molecule of a poison that would kill the world. The apocalypse began with a breath, and your breath tonight is the continuation of that same exhalation. You are breathing out carbon dioxide that the trees and phytoplankton will use to build themselves. The oxygen you take in was produced by some plant, somewhere, some hours or days or weeks ago, released as the plant split water molecules with sunlight. The water those plants split came from the hydrological cycle, from clouds and rain and rivers, and ultimately from the ocean. The cycle runs. The cycle has been running for 2.7 billion years. The breath you are taking right now is connected by an unbroken chemical chain to the first breath that a cyanobacterium exhaled into the ancient Archean sky. The poison is in your blood, and because it is, you are here. Sleep well, breathe slowly. The oxygen will still be there. When you wake, 21% of the air, patient and ancient and extraordinary, the worst thing that ever happened and the best thing that ever happened, the catastrophe that made the world and the gift that sustains it. Good night.